Okay, welcome back. So now we are going to go into the structure and functions of cells. And I know a lot of people don't like going over this material because we're going to be talking about the cellular makeup of structures and tissues of the body. But trust me, it's really important stuff, okay, to understand how cells function. That functioning can then correlate into your understanding of how tissues function. Tissues help you correlate the understanding of organs and organ systems. So it's good to have this basic working knowledge of cells, okay? And there's 70 million cells in your body. Why not get acquainted to them, okay? Structure and function of cells. Cell doctrine. So all living things are made up of cells. Understanding those cells and their function is very important to understanding how our body works. One cell is the smallest form of life. If you think of a cell, okay, a cell has a plasma membrane, just like we have the skin that covers our body. A cell has a nucleus, just like we have a brain that helps to dictate the function of our body. We have what are called organelles inside that cell. Those organelles have different functions, just like the organs in our body have different functions. Okay, so understanding the cell will help you to understand how all those cells function within tissues and organs. Cells come from other cells. So my skin is made of cells, millions of cells. If I stand here, I'm actually rubbing cells off my skin. You could do the same thing. Okay, if I keep rubbing, am I going to rub right down to the muscle underneath? No, okay, because in my skin, the cells at the bottom are producing more cells to create multiple layers of cells to create protection of our skin. Okay, so cells are going to mitose or divide in order to create and replicate and create more cells to provide either protection, or as an organ wears down, cells will be destroyed and more new cells will be created. They say every 7 years that you regenerate most of the organs in your body through that process. And it brings new healthy cells in to replace old dying cells. Okay, when we start to talk about cells, let me zoom in here. Excuse me. The basic structure of a cell will be the plasma membrane and the nucleus. Okay, we're going to talk about more structures than that. Guess what the plasma membrane does? It's just like your skin. It provides a barrier. Now, do we want all substances to sneak into a cell or sneak out of a cell? Absolutely not. We want this plasma membrane or cell membrane to be selectively permeable. It's going to determine what is going to go into it and out of it and regulate that. Okay, it provides a layer of protection. The other thing that it helps to do is regulate the fluids going in and out. Cells need to maintain a certain size and shape for them to function appropriately. If they get too big, just like a balloon that is overexpanded, it can burst. If they get too small, they're too small to provide or perform their functions. So cells want to maintain a certain shape and size. In order to do that, we need to keep a certain amount of fluid inside the cell, okay? If too much fluid leaks in, it can expand. If too much fluid leaks out, it can shrink or crenate, okay? And we'll talk about that in a little bit. So cells, plasma membrane, regulates things coming in and out. A nucleus is going to be the brain of the cells. It's going to help the cells to function, but one of its main purposes, it functions because of DNA, by the way. That genetic code is going to be in each one of those cells. The other thing that it helps the cell to do is to create proteins, okay? There are 3 major macronutrients: carbohydrates, lipids, and protein. Protein's job is to create the structures of the body. Primarily, it has other jobs too. Enzymatic functions, some hormonal functions, but its primary job is to create the structure of the body. The coding for the linkages or the creation of these proteins is in the nucleus. So 2 major jobs. It carries a genetic code, and it also helps to create proteins. Okay. So eukaryotes are the cells in our body with a nucleus. That's what we're going to talk about. We're not going to talk about prokaryotes. This is right in your notes. Okay, prokaryotes are not going to have a nucleus. This is going to be more plant-based products. Okay, so general look at that. Now, the balancing of the fluid, if you look at your next slide, that's where I am. The balancing of the fluid inside and outside the cell is called intracellular fluid on the inside and extracellular fluid on the outside. So this is intra, and intra means inside, extra means outside. So once again, just as I said, it's very important to balance those fluids so we keep the right size and shape of the cell, okay? There's also intercellular fluid, which means if I have two cells, it's the fluid between those two cells. I think you're going to hear more intracellular and extracellular fluid, okay? So one of the big things, I just want to plant the seed as we start to move through this course. One of the big things that's important for you to remember is where sodium goes, and sodium is salt, water goes. Where sodium goes, water goes. So if you go out tonight and have a big salty meal, okay, you're going to ingest a lot of salt. With that ingesting of salt, your body will retain fluid. You get up the next morning, your eyes are all swollen shut, you can't take your rings off, right? Because your body has retained fluid. It's the same thing with fluids going inside and outside of cells. If we put a ton of salt inside of a cell, water will pour in and make that cell grow. If we put more salt on the outside, water will leave the cell and that cell will shrink. Okay, so we want to keep a balance of sodium on the inside and the outside of the cell so we can maintain that proper structure of the cell. Okay, there's more to it than that, but that's just general introduction. Okay, cells are like little people. Okay, we've got a brain, which is a nucleus, a plasma membrane, which is like the skin. They remain small so they can carry on their functions effectively and quickly. Okay, they also have surface specializations. So we're going to go up to the lab if you're taking A&P 1, which you are. We're going to get the microscopes out and we're going to look at these cells under the microscope. When we look at these cells, they will generally have this structure. They'll have the plasma membrane and then a nucleus on the inside. We won't really be able to see the organelles. These are the two main structures that we'll see. What we will be able to see is what's called surface specializations. So we have cells that are shaped like circles. We have cells that are shaped like squares. We have cells that are shaped like columns, okay? And those ones that are shaped like squares and columns can have what's called cell surface specializations, okay? So if we're looking at one of those column-shaped cells, and its nucleus is going to be shaped along the length of it. So it's going to be a little bit longer, okay? Cell surface specializations will be on the top or the exposed surface. We've got microvilli. Do I go into cilia? I don't. Okay, we've got microvilli listed there. And microvilli are like fat little fingers that are on the top of the cell. Now, anytime in the course of our discussions, if I wave my finger in the air like this, and I give you this wavy presentation, that wavy presentation, whether it's on the top of a cell, whether it's in the lining of an organ, is going to increase surface area, okay? So with microvilli, we're going to find microvilli predominantly in the GI tract. Well, why would we want to increase surface area in the GI tract? Because that's where we absorb our nutrients from, okay? So in the GI tract, on the surface of these cells, we have microvilli to increase surface area. So when we eat our food, we can ingest as much of that and absorb it, I should say absorb it, as much of that as possible to get into our bodies, okay? So we have the proper nutrients in our bodies. So one surface specialization is microvilli. Another surface specialization, it's not in your notes, so add this. And most of us are familiar with this, is cilia, okay? Cilia is much thinner, okay? It has what's called an actin myofilament in it. It's much thinner than those wavy projections of the microvilli. And its purpose, for the most part, with an exception in the male reproductive tract we'll get to in the future, is to move substances along, okay? I am sick and I'm creating a lot of mucus in my respiratory tract. Okay, what happens is in the respiratory tract, we have cells with cilia. And what they do is they try to push all that gunk up out of my lungs to clear my lungs. Because if I get that gunk into my lungs, it can fester in there and eventually cause pneumonia. So cilia pushes it up and it actually goes back into your esophagus, into your stomach where you digest it. Gross, right? Okay, so cilia is going to move substances along. Glycocalyx. Glycocalyx, if I look at a cell, okay, glycocalyx is a glycoprotein or a sugar and a protein layer on the outside of cells. Just be a slimy layer that's on the outside of cells. Here would be my nucleus, by the way. Glycocalyx is pretty neat, and it's a great subject of research right now because the glycocalyx helps cells to recognize other cells. It's a slimy sugar layer that makes a red blood cell recognize another red blood cell. So if you were to get a transfusion of the wrong blood type, the glycocalyces would not recognize each other, and your body's immune system would start to kick into play. Okay, so glycocalyx makes you recognize other cells that are either yours or not, okay, and your body responds to that. The interesting part of the research for this right now is that glycocalyx changes in cancerous cells. Cancerous cells can keep modifying it, so guess what your body can't do? Your body's immune system can't say, hey, that's a bad cell, I need to kill it off. Your body's immune system says, oh, I don't recognize that, it must be okay, and it keeps changing and modifying. So it's a pretty neat area of research right now, okay? So cell structure reflects its function. We'll get more into that when we get into tissues, okay? Different types of cells. I'm going to zoom in on this picture just to give you a little introduction. Erythrocytes, known as red blood cells. Okay, they're biconcave. They look like, and some of you may know this, some of you may not, little Werther candies, those little butterscotch candies. If you don't know what they are, go buy some, okay? And what that does is, once again, we're getting an increase in surface area, okay? Nerve cells can be one of the longest cells in the body. Your sciatic nerve is from your gluteal region down to your toe. When people get sciatica, they can get pain all the way down their leg. It's because of that nice long nerve cell. Okay, epithelial cells, we'll get into that, that's covering and lining, okay? Muscle cells, a lot of different muscle cells, those stinking fat cells that everybody wants to get rid of, okay? But they do have an important part in our body. Macrophages help to fight diseases. Macrophages roam through the body looking for stuff to eat up. Anything that's foreign, or if we have tissues that are being broken down, macrophages can eat that up as well. Macrophages are one of the most important part of our systemic immune system. Okay, plasma membrane. You can see that picture there, plasma membrane. If you can't see it really well, you can absolutely pull up a picture online of it. It's made up of what's called a phospholipid bilayer, okay? Phospholipid bilayer. I'm going to draw you a picture in just a second. But there's also cholesterol. There are these little spears of cholesterol that helps to strengthen it. And then you can see crossing that phospholipid bilayer is a protein channel. Okay, those protein channels are super important. The protein channel is the cell's way to talk to its external environment. Those proteins are the cell's way of talking and interacting with the external environment. It creates channels, it creates a receptor for hormones, okay? It creates an area for cells to attach to other cells. So that protein is going to be really important for many, many reasons. When you start to talk about pharmacology, okay, those protein channels are going to be one of the main things that helps medicines work, okay? So lipid bilayer, let me bring you back out, excuse me, to this screen. So if you can imagine, so here's our cell. What I'm going to do is take this little section here and blow it up really big. We're going to talk about the whole plasma membrane, but I just want to look at a segment of it for right now. Okay, so I'm going to take this section and blow it up. And what happens in that section, we have those protein channels, we have cholesterol, but what I want to talk about right now is the phospholipid bilayer. Okay, the phospholipid bilayer is created of phosphorus and lipids. And phospholipid is almost like a double-sticked lollipop. Okay, it's almost like a double-sticked lollipop. There is the phosphorus head, and then there are two fatty acid chains. Okay, so phosphorus head, two fatty acid chains. Now it's a phospholipid bi, meaning two layers. So what happens is we have two of these phospholipids. Okay, and those fatty acids are pointing towards each other. And if we're looking at that little section of the cell membrane, we've got a whole bunch of these, okay? Okay, all the way through the cell membrane. Okay, all the way through the cell membrane. Now, if we think for a minute, if we pour oil and water together, they separate. So this is a brilliant design because the center portion, which are the fatty acids, are towards the center, okay? So that fatty polarized part is towards the center so it can't interact with fluids, but it gives a protective layer. And then the phosphorus is on the outside. And the phosphorus interacts very well with water. So we've got this nice interacting outer phosphorus portion that's dealing with the water inside of a cell and with the water outside of a cell. And then we've got this lipid portion, which is in the center, which helps to provide protection, but it doesn't interact with that water. Okay, so this is a brilliant design. Provides protection in an area that can interact. Okay, now this goes all the way around. Invested through this are those protein channels. Okay, so that's not going to work. Are these protein channels? And you can see the picture right in your book. Okay, this is all filled in, protein channels. And what these protein channels do is help to determine what is going to get into and out of a cell. Okay. Substances can definitely leak through this phospholipid bilayer, but they have to be tiny. Oxygen can leak through, as a good example. Water can sort of sneak its way through. But larger molecules have to go through these protein channels. Okay. So lipid bilayer or phospholipid bilayer helps to create a semi-permeable membrane. Okay. Membrane proteins. So there's different types. I want you to understand in general, I went through this is in the notes, okay? There's integral that's firmly inserted into the membrane. It helps to transport substances across. We'll kind of get through these as we go through different. I don't want you to just memorize these to memorize these. I want you to understand the concept of these proteins crossing through the cell membrane. Okay, and as we get to these proteins, I'll explain their function to you. So I'm going to list these off in the notes. Let me back out a little bit. Okay, so those transmembrane proteins help to transport, help to send signals. Okay, there's a picture of it. Okay, it's also a receptor for transduction of signals. This would be, for example, hormones. Hormones are a ligand system, L-I-G-A-N-D, can attach to it and send a signal through. Okay? Attachment of the structures of the cell or the extracellular matrix in the cytoskeleton. Okay? And then there's a picture of them again, that phospholipid bilayer. Okay? Now, membrane junctions. So we're talking about all of these cells and the structures of the cells, but we've got to remember that these cells will help each other perform a similar function. For example, in the GI tract, there are areas of our body that provide front lines of defense. Your GI tract is a great example. And what your GI tract has along it are what are called tight junctions. So when we look at the cells of the GI tract or the gastrointestinal tract, they are held tightly together with tight junctions. And those tight junctions do not let all substances through. What happens is the cells of the GI tract lining it specifically pick what can pass through. Okay, so tight junctions help to keep out infectious organisms. Think about all the food that we eat that probably has bacteria and viruses and, oh, God, scary thought. These tight junctions between cells help to keep those substances out, okay? Desmosomes, we're gonna go further into this, okay? So I'm kind of giving you a general overview and we'll talk about it more specifically. Desmosomes are almost like cells have been sewn together. So if you can imagine I quilt a whole bunch of segments of material together. That quilt is held together with almost a stitching, well not almost, a stitching like, or a stitch, I'm sorry. That same stitching like presentation is what desmosomes are. It helps to hold them together so those pieces of fabric don't fall apart. The junctions in the skin are desmosomes. Okay, what they do with their primary job is to help hold cells together so they don't separate apart. Gap junctions are really cool. They're little connections between cells that allow substances to pass through them. For example, the signal of the heart happens because of the movement of ions through these gap junctions. Okay, so 3 different connections between cells. Okay, so tight junctions are going to form an impermeable barrier. These are going to be important in the gastrointestinal tract to help keep stuff out that we don't want in there. Okay. When we look at this, of these tight junctions, you can see they're really welded together. And you could look at this in your book. If you can't see this, let me move in closer actually. Whoops. There we go. Okay, these tight junctions are going to really interlock and hold these cells together. So that way, the only way things can get through is through the cell itself. So there are proteins that live on these cells that will let substances pass through. And those tight junctions will keep anything out that we don't want to sneak in there. Okay. Desmosomes are those rivets or spot welds that help to anchor cells together. Once again, these are important when we're talking about the skin. Hold cells together just like the segments of a quilt in a stitch-like kind of presentation. So that way the cells don't fall apart or move apart from each other. Okay. Excuse me. And there's an example of a desmosome there. You won't need to know what they look like. You need to know in theory what they do. And that's part of what I do with this class, is try to give you visuals, because there's so many different type of learners, to give you an idea of what things look like. But I would never put this particular picture up and ask you, is this a desmosome? I want you to know in theory what a desmosome does. Okay? So gap junctions are almost like pores, okay, that allow substances to pass through. Once again, the ions through cardiac muscle, which we'll get into ions in chapter 2, where we got into ions in chapter 2. Ions are going to help to perpetuate the contraction of the heart muscle. Same thing with smooth muscle cells. Okay, and you could see in that picture, looks just like a little channel or a pore. Okay. So different types of transport. We have passive transport, we have active transport, and actually underneath active transport, you can exclude exocytosis and endocytosis. Now, passive transport is going to require energy. This is the difference between passive and active transport. Passive transport is going to be substances moving of their own accord, no energy is used, okay? Energy in the body that we're talking about is going to be adenosine triphosphate. Adenosine triphosphate helps to give energy to structures in the body. So during passive transport, no ATP will be used, okay? During active transport, we will use ATP, okay? So energy is used during active transport, okay? ATP is used, okay? There we go. So passive transportation, examples would be diffusion and osmosis. So diffusion, okay, just let me see if I have, okay, sorry. I wanted to make sure I had no other notes about that. Diffusion is going to be simply the movement of particles in a fluid until they're evenly distributed throughout a fluid, okay? There's no pumps, there's no cells involved. It's just passive, you know, just like if you've ever noticed with a party. You walk into a party, and instead of everybody being clumped in one area of the room, people spread out. There you go. There's passive diffusion. Same thing happens with cells in a solvent, okay? They will move unless, you know, they settle out because of gravity or whatever. But for the most part, they will diffuse throughout that solvent, okay? Osmosis is going to be the movement of water against a selectively permeable membrane. I think in this case, a picture is worth 1,000 words, okay? If we look at the next picture, so we can move, don't forget, substances can move either through that plasma membrane or through the protein channels, okay? So through plasma membrane and through the protein channels. But what we're talking about with diffusion and osmosis is just in general what they are. Okay, so diffusion is going to be the even distribution of a solute, which is the substance in a solution, and into a solvent. Okay, so if we're talking about salt water, we're talking about salt water. Salt is the solute, water is the solvent. When we put that solute of salt into the solvent of water, we're going to have a solution. Okay, so that's what we're talking about with that. I'm going to zoom back in. Now, here's a picture, and I'm sorry, the links are not working for the movies. So when you see that in the notes, I apologize. Okay, so this is diffusion. This is passive diffusion, where we put cells into a solvent and they spread evenly throughout the solvent. And what happens is that little bit of movement that cells have, because they do move a little bit, will actually interact with the cells around them, and they keep bumping into each other until they're evenly spaced throughout that solvent. Here's an example of osmosis, and some of you may have done this in basic chemistry. If we have a U tube, and we have a selectively permeable membrane at the bottom, so if you look, it says membrane. And I'm saying selectively permeable because the solute cannot pass through it, okay? The solvent can, but the solute cannot. So that's what we're talking about here. So if we put more solute on the right side, if you look at that U tube, and we put more solute on the right side than the left, what will happen is water will start to go where there's more solute. Water is going to try to balance out that solution and try to evenly distribute the particles of solute in it. So in order to do that, it will actually go higher in the U tube, okay, on the right side, and sort of almost fight gravity. But that's because of that distribution of the solute in the solvent. Excuse me. Active transport is going to expend energy. Now, don't forget, ATP, adenosine triphosphate, will actually get rid of a phosphate and give it to a structure and turn into adenosine diphosphate. Okay? Adenosine triphosphate gets rid of a phosphate. That's how it provides energy to structures, and it'll turn now into ADP. Okay? So, all of these pumps will use ATP or energy. Exocytosis sucks stuff out, and I'll show you a picture in just a second. Endocytosis sucks stuff in. I think a great example is macrophages. Okay, we have cells in the body who are the cleaner uppers of the body. If there's a bacteria, if there's an infection, if there are dead cells, These macrophages are big, super huge cells that will go, not super huge, but bigger cells that go to that area and I'll suck it in. Okay, they pull that stuff in and they have enzymes that'll then break that stuff down and break it into parts to get rid of it. Okay, that's endocytosis. Exocytosis is when cells sort of spit stuff out. Okay, I should have said spits stuff out. Exocytosis. So if we're making those proteins that I talked about, the nucleus senses we need more proteins in an area, creates more proteins through many processes we'll talk about in the cell. It's going to have to send that protein out of the cell via exocytosis, okay? So we can spit stuff out or bring it in. Both of them are active. Now, there are protein pumps that work as transporters, or I guess you'd say co-transporters. There's a sodium-potassium pump that's going to be the important one that we'll talk about. Okay, sodium-potassium pump is going to help regulate the amount of sodium and potassium on the inside and the outside of the cell. Here's an important note. Sodium, you might want to write this down, is the predominant ion on the outside of a cell. Okay, sodium is the predominant ion on the outside of the cell. Potassium is the predominant ion on the inside of the cell. Okay, potassium is the predominant ion on the inside of the cell. Why is it this way? This is how our body regulates ions to keep the right charge on cells. We'll talk about that in a little bit more specifically. Okay, so sodium is, oh, it's right in the next set of notes. Stand up or read ahead, sorry. Sodium is mostly on the outside, potassium is mostly on the inside. That's an important physiological thing to know. Okay, here's some pictures. Once again, active transport. You can see it's turning ATP into ADP, okay, using energy. Endocytosis and exocytosis, okay, bringing stuff in or spitting stuff out. Another thing to remember when it's spitting stuff out or it's bringing stuff in is it will create a membrane around that structure or that substance when it's inside of the cell. And that's what's happening here. You can see that membrane will actually attach to the membrane or the plasma membrane of the cell and let stuff out. If we're bringing stuff in, it'll sort of invaginate or bring in that cell membrane and enclose that structure off as it comes in. Okay, endo and exocytosis. Good. And here's an actual picture of it. I thought that was kind of a cool little picture. Okay. Endocytosis, also known as phagocytosis. Okay. Macrophages and some white blood cells, such as neutrophils, will do this, bring those structures in. First, little pseudopods are really cool little structures that almost look like hands that are reaching out, and it feels for those substances. It actually smells it first through a process called chemotaxis, but that's getting beyond what we need to know. It sticks these little arms out called pseudopods to bring stuff in. Okay, phagocytosis. Okay, another explanation with another picture. Okay, sodium-potassium pump. Let me just see what's next. Okay, sodium-potassium pump. You know what, actually, I'm going to take a little break. I want you to take a break too. Go get a cup of coffee, and I'm going to bring it back out. Okay, go get a cup of coffee, and I will be back here. We'll restart up the next of it. Put on your thinking cap because the next stuff is a little difficult. Okay, thanks. I'll be right back.